Hello dear students, myself Vishal Naik, working as an assistant professor in Department of Chemistry, MPSC College Panvel. So our today's topic of video that is based on analytical chemistry, okay, and that particularly in analytical chemistry we are going to study optical methods. So we already discussed one instrument, that is the flame emission spectroscopy. So in optical methods, next instrument that is we have to study, that is atomic absorption spectrophotometer. So in this particular video we are going to discuss principle of atomic absorption spectrometer, then instrumentation of atomic absorption spectrometer, then application of atomic absorption spectrometer, and last one, that is the limitations of atomic absorption spectrometer. So what is the principle behind this atomic absorption spectrometer? So as we know, in a flame emission spectrometer, we aspirated sample that is contain a metal ion. Okay, vaporization takes place, then atomization takes place, and molecule absorbs the energy, and molecules absorb, by absorbing that energy, molecules goes from ground state to excited state. Okay, but always remember that only small amount of molecules get excited and maximum amount of atoms are remain in a ground state. So FES is based on the emission radiation, that is come when molecule emit radiation that are determined in flame emission spectrometer. So the principle remains same as flame emission spectroscopy, okay, but only a single change in atomic absorption spectrometer. Atomic absorption spectrometer is only focused on ground state atoms, not an excited state atoms. Okay, so this is general principle of atomic absorption spectrometer. So atomic absorption spectroscopy is the study of absorption of radiation by the ground state atom in a gaseous state. Okay, so in gaseous state is there, we have to burn that particular sample, same as FES in a flame. Okay, then this main atomic absorption spectrometer mainly focused on the atoms remain in the ground state. So next part, that is the instrumentation of, so the instrumentation is entirely different as compared to our flame emission spectrophotometer. Why? Because there is slits and all are present, but in this case the most important point or most important part of this instrument, that is the halo cathode lamp or halo cathode cup lamp. What is halo cathode cup lamp? In the next part we actually discuss on that. So halo cathode cup lamp, that cup lamp actually contain a metal cup. Okay, if we have to determine the copper, so we have to take a hollow cathode cup lamp which is made up of a copper. If we have to determine the iron, okay, so we have to take hollow cathode lamp, cup lamp which is made up of an iron. So separate lamps are required for the determination of different elements. Okay, so that particular lamp actually gives a radiation or emit a radiation. Okay, then chopper is used. So the chopper actually chops down that particular wavelength. So different wavelengths are there, but the most intense wavelengths are passed through this. Then here is again a sample sucker, that is the atomizer. We actually know that. So sample is burned in this case. So maximum amount of ground state atom remain in this case. So the radiation comes from this hollow cathode cup lamp. Because if we have to determine a copper, so the radiation comes from that particular hollow cathode lamp is same as that element. Okay, so that particular element only absorbs that radiation. So detector detects only how much amount of sample does not absorb the emitted radiation, absorb radiation. So this particular detector detects and differentiate it, and we get our answer in our atomic absorption spectrometer. Okay, so these are the components of AAS. First is the source of radiation, that is a hollow cathode cup lamp, then rotating chopper, then flame atomizer, that is called as a burner, then monochromator, that is the different type of monochromator, quartz, glass and prism is also used. Then photocell, it is actually intensity of to electric current, converts the intensity into electric current. Most probably or mostly we use a photo multiplier tube in FES also and AAS also. Then amplifier, which amplifies the electric signal, comes from this photocell. Then readout, convert that into a particular power. Then this one is a source of radiation, that is hollow cathode cup lamp. So we already know that this cup lamp is made up of a metal which we have to determine. okay, or the sample solution containing that particular metal ion, we have to determine that. So we have to use that particular metal ion cup cathode lamp. So hollow cathode cup lamp, that is the made up of an element under study. Example, if nickel is to be determined or detected in the sample, we have to use this lamp which is made up of a nickel. Then the cathode is protected in a glass shield. So this is in a glass shield. Okay, so this cathode cup lamp, which is act as a cathode, is protected by this glass jacket. Okay, and a tungsten wire is act as an anode. So this wire is act as an anode. The two electrodes are enclosed in a glass jacket containing an inert gas like neon or argon at low pressure. So this gas actually work here. So neon or argon gas is filled in this particular jacket, particular glass. So when a DC voltage of 300 volt is applied, okay, so when applying this voltage, the inert gas get ionized. So how that hollow cathode cup lamp actually work? So when we apply 300 volt energy through this cathode and anode, the inert gas, that is neon and argon, actually ionize, and positively charged argon or neon is attracted towards this cathode. So in this case, so argon or neon, positively charged ions get formed by applying this 300 volt energy. So these positively charged ions attracted towards this cathode. Okay, so this is the most important point. So these fast moving ions actually hits that cathode, and the electrons or the energy or that particular hitting is called as a sputtering. So due to that sputtering, the metal cup, actually this is a metal cup. So this metal cup collide with high energy which is produced in this particular entire part, and what happens? So electronic excitation takes place. Okay, so gaseous ions attract towards this. So this cup, actually smaller amount of metal ion get detached from that metal ion, okay, due to high energy, and that ion or that particular ion get, or electrons present in that ion get excited, and that excited ion emit that radiation. That emitted radiation is same as absorb, as we already know in the principle. Okay, so that particular light, emitted light, is act as a source. So neon or argon gas actually work here, okay, which activates this cup cathode, and emitted radiation get passed through this quartz window, which is focused on a next part, that is our burner, which contain a sample solution. So this is in a diagram, and this is the original hollow cup cathode lamp as we seen in this diagram. Then flame atomizer or the burner. So this is particular burner that is used in atomic absorption spectrometer. So it consists of a graphite tubes open at both ends. Okay, the tube is around 5 centimeter long, surrounded by a metal jacket, and water circulation is there to control the heating. Okay, 2 electrodes are fitted at the end of the tube, and the steam of the gas like argon and neon flows into that. Then that particular argon and neon gas prevent the oxidation of the graphite material. Okay, the beams that comes from the source of radiation, that is emitted radiation, pass through this line. Okay, go towards the detector. So in this particular part, flame is there where metal ion get ignited or metal ion get flamed. Okay, lesser amount of metal ions are get excited, but maximum amount of metal ions present in that part for some time. Okay, so lifetime is very much short, but they are present in that time. Okay, present in that part. So that particular sample is present in the ground state, absorbs the radiation which comes from this one. Why? Because if this one is a nickel, cup cathode lamp is nickel lamp. Okay, and sample contain a nickel. So nickel absorbs the energy which comes from that lamp and goes towards this detector. So without a metal ion, without a sample, 100% intensity goes towards the detector. But when samples absorb the energy, only few amount of radiation goes towards the negative. So by differentiating that particular detector detects that how much amount of sample or how much amount of sample is present and absorbs the energy. So this is the role of atomizer. It actually work at 2000 to 3000 degrees Celsius, where maximum amount of sample get ashed down. Okay, so it atomizes into different part, and that atoms actually absorbs that energy. So next part in this, that is the application of AAS. So application of AAS and FES both are same. That is quantitative and qualitative. So in a qualitative analysis, different type of metal ions present in a different sample are determined by using AAS. Particular element is detected, then estimate trace metal in biological samples, detect toxic metals which are present in a food products, estimation of sodium, potassium, blood serum and lead in a petrol. Then quantitative analysis same as FES. That is, there are three methods. First is the calibration curve method, where we aspirated different sample, we prepare a series of standard solution. One by one we aspirated into a flame, determine the absorbance, because we know the concentration, we determine the absorbance. By using that, we plot this calibration curve method. So then we aspirated our unknown sample solution, so we get absorbance of particular sample, and then by using that absorbance we determine the particular concentration of the metal ion present in that solution or sample solution by using calibration curve method. In standard addition method also, X remains same. There is an unknown sample solution, amount of unknown solution remains same. So we again prepare a series of standard solution. Same amount of unknown is added, and then the standard is added in a variation in amount. And one by one we aspirated into a flame, and by using that aspiration we determine the absorbance. Okay, in a flame emission spectrometer we determine the emission intensity. In AAS we determine the absorbance. So again, by using this plotting, by plotting the graph of that, by extrapolating that, we on this side we get our concentration of our unknown sample solution. Then last method, that is the internal standard method. So here, instead of emission, absorption is there. So absorbance of an analyte, then absorbance of internal standard, the ratio is taken. Okay, and we plot that ratio against our concentration of standard solution, and by using this graph also we determine the what amount of metal ion present in that particular sample solution. And the last but not least, the limitations of atomic absorption spectrophotometer. So separate lamp is required. If separate lamp is required, then the costing of that particular instrument is very much large. But nowadays multi-channel lamps are used to reduce the cost. So this one is in the general limitation of our AAS, that is the separate lamp is required. Then metal like lithium, aluminium, titanium, tungsten, molybdenum are not normally analyzed in AAS. Okay, because they dissociated by nitrous and acetylene flame. So if dissociation takes place, so we cannot determine the actual amount by using AAS. Then interference due to anions like phosphates and other are also there. Then if two elements absorb same radiation, so it determined as a single one. So we have already studied in FES. Then role of solvent. So aqueous solutions are more difficult to atomize than non-aqueous ion. So as compared to aqueous solution, non-aqueous solutions are also used. Then low ionization potassium metals, moderate temperature flame is used. So this is all about the atomic absorption spectrophotometer. Thank you.